Yeah. So students, here if we see first types of ovules. Types of ovules, we have 6 types. First and basic type is orthotropous ovule. In orthotropous ovule, what we have discussed, micropylar end, chalaza and funicle, they will be in the straight line. Okay, what are the examples beta? Cycas, pipper and polygonum. In 80% of angiosperms, what type of ovule will be there? Inverted ovule, which is inverted to 180 degrees. Okay, this is present in 80% of angiosperms. Okay, hemianotropous, where the body of ovule is at the right angle to the funicle. Example is ranunculus. Okay, Campylotropous, where ovule is slightly curved. Ovule is at the right angle to funicle, but it is slightly curved. Examples include Leguminosae and Cruciferae. Next, here you see Amphitropous, where ovule and embryo sac both are slightly curved, and ovule will be seen as what beta? Horseshoe shaped ovule. Okay, examples is Lemma, alisma and poppy. Okay, Sarcinotropous ovule, where ovule is completely revolved 360 degrees. Okay, and funicle is all around the ovule. Okay, examples include what? Plumbaginaceae, Opuntia, which belongs to Cactaceae family. Clear? These are the examples. What is primary thing that you have to remember from types of ovules is each type, what is the exact position of micropyle, chalaza and funicle, and examples of that. Enough beta. Okay, yeah. Just a second. Yeah. Now, megasporogenesis. Okay. Before dealing with megasporogenesis, I will give you a quick idea of the female reproductive structure. In female reproductive structure, let us take a single carpal unit. Okay, we all know a single carpal unit will have what ma? Stigma, style, and the basal bulged portion is what? Ovary. This basal bulged portion, ovary, will consist of what? Ovules. It can be single ovule or many ovules. This arrangement of ovules inside the ovary is called as placentation. Okay, so ovule post fertilization will turn to what? Seed, and this ovary will turn to what? Fruit. Okay, so single ovule, types of ovules we have learnt. Each ovule, if I have to consider a anatropous ovule, as most of the angiosperms will have anatropous ovule. If I have to consider an anatropous ovule, inverted ovule, the cells which are present in this ovule are called as what cells? Nucellus cells. Okay, I will not draw an embryo sac here only. So, what are these cells called as beta? Nucellus cells. Fine, right? Nucellus cells. These nucellus cells, one of the nucellus cell in the micropylar region which has dense cytoplasm and nucleus, which is large, which has dense cytoplasm and nucleus, one of the nucellus cell in the micropylar region will get differentiated to megaspore mother cell. Okay, it gets differentiated to what? Megaspore mother cell. This megaspore mother cell will undergo reductional division or meiosis. Okay, this will undergo a reductional division or meiosis, wherein single megaspore mother cell will undergo meiosis. After meiotic division 1 and meiotic division 2, how many daughter cells are formed? 4 daughter cells are formed. As such, 4 megaspores are formed beta. Okay, which is a linear tetrad. Yes, Raghav, what is your doubt?
But only one survives.
Yes, not survives, it is functional. So, it is a linear tetrad of what? Megaspores. Megaspores are haploid. Megaspore mother cell is diploid. Okay, so consider this as chalazal end and this as micropylar end. Okay, nucellus at the micropylar region is differentiated to megaspore mother cell. After the formation of a linear tetrad, 4 cells are linear tetrad, right? Chalazal end and micropylar end is clear to you. 3 cells towards the micropylar end will be degenerated. This will be degenerated, and 1 cell which is towards the chalazal end is functional. Listen carefully. From here only embryo sac will be formed. You listen. Okay. So, functional megaspore is at what end, beta? Functional megaspore is close to chalazal end. Till here the story is clear, no? The story of megasporogenesis till here is crystal clear, right? You know what is the meaning of megasporogenesis? Production or formation of megaspores from megaspore mother cell is called as megasporogenesis. So, till here the formation of functional megaspore is clear to you. Okay?
This reduction is called megasporic meiosis, something like that.
Sporic meiosis. Okay. So, here you see at the micropylar end megaspore mother cell, after differentiating meiosis 1, meiosis 2, 4 tetrad is formed. Functional megaspore which is towards what end? Chalazal end. And here you see this chalazal end functional megaspore, chalazal end micropylar end. Okay. This will undergo first mitotic division. Sequential mitotic divisions will happen. Sequential mitotic divisions. So, before learning this, one thing should be clear to you, like the phenomena where a single megaspore which is towards chalazal end is functional is addressed as monosporic development. Monosporic development of megaspore or embryo sac formation. As such, you will have three ways, three types. One is monosporic, second is bisporic, and third is tetrasporic. Okay. Here monosporic is also called as polygonum type. Monosporic is also called as what type? Polygonum type, which is most common. That is, from a single functional megaspore the embryo sac formation takes place. Okay. Embryo sac formation occurs in three ways. One is monosporic development, that is from a single megaspore, okay, which is also called as polygonum type. Second is bisporic, where two megaspores will be functional and two will be degenerated. Best example is allium. Fine. Next is tetrasporic, where all the four megaspores will be functional and they are involved in the formation of embryo sac.
All of these will be closer to the chalazal end.
No, they are from chalazal end to micropylar end.
But none in micropylar. None.
They are present like this beta, it is a linear tetrad.
Yes, no. So bisporic will be there. So
bisporic 2 megaspores of chalazal end. Why it is like that I will tell you. Now remember the point, why is it so I will tell you. Okay. So here tetrasporic type of embryo sac development, here all the four megaspores will be functional. Examples you can take adoxa and plumbago. Clear, right? Next. Abdul, done, na?
how many abnormalities observed in bisporic and
Nothing like that. It is a common phenomena where embryo sac is formed from 2 megaspores, embryo sac formed from 4 megaspores. But as polygonum type is most common in the angiosperms, we learn about polygonum type only in most detailed way. So here the functional megaspore which is at the chalazal end, after first mitotic division, it forms 2 megaspores. Okay, it forms 2 megaspores, one at the micropylar end and one at the chalazal end. Okay, and second mitotic division, again this forms 2 cells at, sorry, this is considered as chalazal end because functional megaspore, right, chalazal end and micropylar end. And here again chalazal end, micropylar end. One more mitotic division, third mitotic division, it forms 4 cells at the chalazal end, again 4 cells at micropylar end. Now what happens is, one cell from the micropylar end One cell from the micropylar end will come to center. One cell from chalazial end also comes to center, forming a central cell or polar nuclei. Okay, and 3 cells which are present at the micropylar end are called as egg apparatus. 3 cells which are present at the micropylar end are called as what? Egg apparatus, where it has 1 egg cell and 2 synergids. And 3 cells which are present at the chalazial end, these are called as what? Antipodals. Okay. Now you see, now you have understood, no, this is the complete embryo sac. Embryo sac. Fine. This is formed from 3 sequential mitotic divisions. 3 sequential mitotic divisions from a single megaspore. This is what type of development, beta? Monosporic type of development, embryo sac. Monosporic type of embryo sac development. Fine, right? Now we will study the structure of embryo sac in detail. The story will be very interesting. Formation of megasporogenesis might be, microsporogenesis might be very interesting. So here you see embryo sac which is embedded in the nucellus cells. All the cells present in the ovule are nucellus only, no. One nucellus got differentiated to megaspore mother cell, that undergone meiosis, 4 megaspores, from that one functional megaspore, 3 mitosis formed an embryo sac. Now this embryo sac is embedded inside what? Nucellus cells of the ovule. Till here it is okay, no? Now you see the structural description of embryo sac. First we will look at the micropylar end. At micropylar end I told you what, beta? Micropylar end you have egg apparatus. Egg apparatus wherein 2 synergid cells are there. Synergid cells are also called as addressed as helper cells or cooperative cells. Okay, they are also known as synergid cells, helper cells or cooperative cells, because mostly the pollen tube entry will be from the micropylar end only, and synergid cells will have filiform apparatus. Okay, so pollen tube will be entering through one of the synergid, not both. Through one of the synergid it will enter and the filiform apparatus will be ruptured there. Okay, and that is how it enters the egg cell. Egg cell will maintain the polarity. Egg cell is close to the micropylar end. Okay, the nucleus of the egg cell is close to the micropylar end. It will maintain that polarity.
How does it maintain polarity?
How does it maintain that? It is waiting for the male gamete. So it is close to the micropylar end. Understood? Next at the center you have polar nuclei. Okay, second male gamete from the pollen grain, it will come and fuse with what? Polar nuclei. This is called as triple fusion. Okay, and this forms primary endospermic nucleus. And 3 antipodals are present at the chalazial end. Okay, these antipodals will be degenerated after fertilization. Clear? Antipodals will be degenerated after fertilization. Clear, right? Clear with the structure of embryo sac? Next, pollination. Types of pollination you see. We have 2 types of pollination. One is self-pollination, second is cross-pollination. What is self-pollination? Self-pollination indicates that the pollen grain from the same plant, if it is germinating on the stigma of the same plant, of different flower might be, or on the stigma of same flower of the same plant also, is considered as self-pollination. Self-pollination is of 2 types, autogamy and geitonogamy. Autogamy means pollen grain pollen grain germinating on the stigma of same flower, same flower. Okay. Geitonogamy, where pollen grain, yeah, different flowers of the same plant. Pollen grain germinates on the stigma of different flower same plant. Okay. So, this is functionally cross pollination, but genetically it is self pollination. Geitonogamy is functionally cross pollination, genetically self pollination. Okay. Next, cross pollination is also addressed as xenogamy. Xenogamy means pollen grain germinates on the stigma of different plant and different flower. Okay. Now you see, what is the adaptation of a plant to ensure the self pollination? Okay. To ensure self pollination to happen, what are the adaptations of a plant? Okay. Take down. Adaptations of a plant to ensure self pollination. Okay. First is bisexuality. What is bisexuality? That a single flower has both androecium and gynoecium. Bisexual, can I also call it as monoecious condition? Next is homogamy. Which means both stamen and pistil matures at the same time. Okay. Both androecium and gynoecium matures at same time. Male and female reproductive structures mature at same time. Third is by having clistogamous flowers. Clistogamous flowers. There are few plants like viola, which is also called as common pansy, oxalis and commalina. Okay. So, these plants, these clistogamous flowers will never open at all. The flower will be in the bud stage, it is bisexual and it will never open at all. When the flower is not opening at all, definitely the pollen grain which is released from that anther will be germinating the stigma. So, it conducts what pollination? Bud pollination. Okay. These are the adaptations of a plant which ensures self pollination. Yes. Good, that is the problem. You have good memory but you will never revise. Just revise and see, Raghav. Here we go, okay. Self-pollination and cross-pollination I already told you. Autogamy, in the same flower you see. Geitonogamy, different flowers of the same plant. Xenogamy, and different flowers of different plants. Last slide you see, these are cleistogamous and chasmogamous flowers, wherein some plants like viola, common pansy, oxalis and commelina produce two types of flowers. At the base region, okay, at the base region, cleistogamous flowers are there. These cleistogamous flowers will never open and they will conduct only autogamy. But chasmogamous flowers which are above, they will conduct both autogamy and geitonogamy. The flowers are open here, they will conduct both autogamy and geitonogamy. Clear, right? So, with this we have dealt with what today? Types of ovules. Next is megasporogenesis. In that, embryo sac development, 3 types. Monosporic, okay, you write first. You write first, after which I will discuss. Completed. Completed. So, what we have discussed today, beta? Types of ovules, okay, and then we have discussed about megasporogenesis, 3 types of embryo sac development: monosporic, bisporic and tetrasporic, embryo sac structure, okay, after which we started on discussing pollination, because after pollination only we will discuss pollinating agents and double fertilization. So, in pollination it is of 3 types, 2 types majorly, self pollination and cross pollination. Self pollination again it can be through autogamy and geitonogamy. What are the plant adaptations to conduct autogamy mode of pollination we have discussed, and geitonogamy as well, okay. And then xenogamy, that is cross pollination, involves or it requires pollinating agents. Pollinating agents can be abiotic and biotic. So, about the pollinating agents, that is biotic and abiotic pollinating agents, and then about double fertilization, artificial hybridization we will look in the next class. Okay, till here so everything is clear, no ma? Yeah. So, that is all for today's session. Just keep on revising this, practice the objective of DPP, NCERT beginners till Excel, okay, from this till here wherever we have completed. Okay, yeah, thank you.